Assalamu alaikum wa rahmatullahi wa barakatuh. Today we're going to explain the second step of protein synthesis, translation. At the end of the video, you will recognize that translation is the mechanism by which mRNA serves as a template for the sequencing of amino acids for a polypeptide that takes place in the cytoplasm. Specify the respective roles of tRNA and ribosomes in protein synthesis. Compare mRNA to tRNA, specify the steps of translation, and learn how to use the genetic code and identify the different types of codons, initiation codons, stop codons, and redundant codons. Protein synthesis occurs in two steps, transcription and translation. We already discussed the first step, transcription, in the previous activity, where a copy of DNA will be created, called mRNA, and in this activity, we will discuss the second step, where we will learn how the peptide chain is produced. Every mechanism requires certain tools to be accomplished. Let's examine the tools used through this mechanism together. A close look at the rough endoplasmic reticulum allows to identify mRNA associated to 20 nanometer granules bound to it. These are the ribosomes. They are the site of protein synthesis. Let's have a quick look at the tools of translation. Translation involves the mRNA strand, tRNA, and the ribosome. A ribosome is made up of two subunits. The large subunit and the small subunit. The small subunit is the structure that the mRNA will bind to. The large subunit includes two main chambers, A site and P site, where it uses them to decode the mRNA and build up the peptide molecule. The coded message carried by mRNA to ribosomes needs a translator capable of understanding two languages, the nucleic acid language and the proteic one. This translator is the transfer ribonucleic acid, or tRNA. It is a short single-folded chain and has two attachment sites, amino acid attachment site and anticodon attachment site. Keep in mind that mRNA is also like tRNA, a single-stranded molecule, but it carries the codons, while tRNA holds anticodons complementary to the codons found on the mRNA. Before we start examining what happens during each phase of translation, we should keep in mind the elements of translation, which are mRNA, ribosomes, and tRNA. Translation includes three steps: initiation, followed by elongation, and finally termination. During initiation, an initiator tRNA helps the two subunits of the ribosome to come together around the target mRNA. The initiator tRNA carrying the first amino acid methionine occupies the P site of the ribosome and binds to the start codon AUG. During elongation, a new tRNA occupies site A of the ribosome and binds to the second codon on the mRNA. Then a peptide bond is formed between the two amino acids. The ribosome is then translocated, thus liberating site A for a new tRNA to occupy. During termination, the ribosome reaches a stop codon on the mRNA. Then synthesis stops and methionine is liberated. Finally, the ribosome dissociates into its subunits. We are given the following mRNA sequence. By referring to the genetic code table, we obtain the following peptide sequence. We notice that the start codon AUG encodes for amino acid methionine. But we also notice the presence of different codons that encode for the same amino acid, like UC, and UCU. They both encode for serine. These codons are described as redundant codons. And finally, we notice UGA codon, which doesn't encode for any amino acid. Then it is described as a stop codon. How many stop codons can you see in the genetic code table? Yes, there are 3 stop codons, UAA, UAG, and UGA. Now together let's watch an animation that describes the different stages of translation mechanism.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Now, after watching the video, let us check our knowledge about this mechanism. Translation is the first step of protein synthesis, second step of protein synthesis, a process that produces the protein molecule, or a process that hydrolyzes the protein molecule. So here we have 2 correct answers: B, second step of protein synthesis, and a process that produces the protein molecule. Translation takes place in the cytoplasm, nucleus, or mitochondria. Yes, it is true. It takes place in the cytoplasm, so answer A is correct. The factors needed for translation are DNA polymerase, ribosomes, tRNA, mRNA. Yes, there are 3 correct answers: B, ribosomes, C, tRNA, and D, mRNA. Translation includes 2 steps, 3 steps, or 4 steps. Yes, correct. Part B, 3 steps. The first step of translation is elongation, termination, initiation. Yes, part C is correct. Initiation. Ribosomes are made up of 2 subunits of equal sizes, 2 subunits, one small and the other large, or 3 subunits of equal sizes. The correct answer is B, 2 subunits, one small and the other large. To sum up, translation is the second step of protein synthesis that takes place in the cytoplasm. Translation involves the action of ribosomes and tRNA. tRNA, or transfer RNA, is the molecule that is capable of attaching complementary amino acids to the codons of the mRNA. Ribosomes are the site of protein synthesis that attach to the mRNA strand and allow the tRNA to translate it. Translation includes 3 steps: initiation, elongation, and termination. During initiation, the start codon AUG is recognized and the initiator tRNA carrying the start amino acid, methionine, binds to it. During elongation, amino acids are brought to the mRNA by tRNAs, one after one, and thus the peptide chain will grow longer. During termination, a stop codon is recognized by a protein release factor, and the translational apparatus comes apart, releasing the synthesized polypeptide. In the genetic code table, there are different types of codons: initiation codon, stop codons, and redundant codons. Thanks for listening.